In the last class we discussed about the electron transport chain and involvement of complex 1 and involvement of complex 2. In that case, complex 1 is involved, 6H plus ions are transported, that one is supporting for the synthesis of 3 ATPs. If the complex 2 is involved, that one is supporting for the synthesis of 2 ATPs, because 4H plus ions are transported by the complex 2 involvement and 6H plus ions are transported by the involvement of complex 1, right? Both the cases there is an electrochemical gradient generation, that generation supporting for the synthesis of ATP, right? After that synthesis of ATP, coming to the next topic is the inhibitors of the electron transport chain. See, these are the inhibitors, right? See, that inhibitors blocks the transportation of electrons. That inhibitors blocks the transportation of electrons. So here there is a complex 1, complex 2 is there, which are involved, either maybe the complex 1, either maybe the complex 2, both these are interact with that coenzyme Q. This one is also called as ubiquinone. Here the flow of electrons from maybe the 1, maybe the 2, the flow is blocked by the amytal. Okay, this is one of the inhibitor, right? Next one is the rotenone, right? Rotenone is another inhibitor. So these are the inhibitors which inhibits the flow of electrons from 1 to coenzyme Q or 2 to coenzyme Q. So the flow is inhibited, the electron flow is inhibited. After that coenzyme Q, the next one is that cytochrome B, right? That cytochrome B, okay, naturally it transports that electrons from B to cytochrome C, right? Here again antimycin is the inhibitor. Antimycin is the inhibitor. Remember, these are the inhibitors which blocks the transportation of electrons, right? After that cytochrome C to cytochrome C1, electrons are transported. See the cytochrome C1 to cytochrome A3. Remember, this is the main area. This is the main area. We know about the cyanide. Some people are taking cyanide, immediately the people will be dead. See, the cyanide acting electron transport chain complex is 4. The cyanide acting electron transport complex or electron transport chain complex is complex 4. So especially the cyanide act on the complex 4, which blocks that electrons flow from cytochrome C1 to cytochrome A3. Here cyanide, again the coenzyme Q and sodium azide. All these are, you see, not coenzyme Q, this is carbon monoxide, sorry. See, this is the cyanide and this is the carbon monoxide, right? See, carbon monoxide, cyanide and the sodium azide, all these are act on that complex 4, especially which blocks the electrons flow from cytochrome C1 to cytochrome A and A3. So here what I'm telling, cyanide is especially act on that 4th complex, no doubt about it, right? See, along with that cyanide, some of the inhibitors also acted. Carbon monoxide is one of the best example. Sodium azide is also one more example, right? So these are the inhibitors which blocks that complex 4. Right, and see, finally there is no conversion. If in any place, if the flow of electrons is blocked, then finally no need of molecular oxygen, because if the electron flow is there, then definitely molecular oxygen function is required. So if the flow is blocked at somewhere within that electron transport chain, right, then there is no need of molecular oxygen. So here that is about that inhibitors of electron transport chain. Remember, here this is the one of the site, amytal and rotenone, right? And next one is that antimycin between that cytochrome B and cytochrome C, right? Here especially these are act on complex 4, right? See here whatever that electron flow from C1 to A3, this is the complex 4, right? So here cyanide, carbon monoxide, sodium azide, these are the inhibitors. These are the inhibitors which blocks the electron transport chain. Right? That is about the inhibitors of electron transport chain. And we know about that complex 5. We know about the complex 5. See, the complex 5 is also called ATP. ATP synthase enzyme. ATP synthase enzyme. That ATP synthase enzyme especially blocked by the oligomycin. Oligomycin is one of the inhibitor oligomycin is one of the inhibitor. That oligomycin act on the complex 5 or ATP synthase enzyme, blocks the ATP synthesis. That is about the inhibitors, right? Yeah, coming to the next one is respiration coefficient. See, respiration coefficient values are very important. Sir, what is respiration coefficient? Remember, during the respiration process, certain amount of carbon dioxide is liberated and certain amount of oxygen is consumed. Then we are doing the ratio between the amount of carbon dioxide is liberated and amount of oxygen is consumed. So that ratio is called as respiration coefficient. The ratio is called as respiration coefficient. Very simple. During the respiration process, certain amount of carbon dioxide is liberated and certain amount of oxygen is consumed. So we are doing that ratio between the carbon dioxide and the oxygen. So that value is called as respiration coefficient. Respiration coefficient. So these values also called as RQ values. These values also called as RQ values. So the respiration coefficient measured by, say, one of the device is there, that one is called as Gannang's respirometer. That particular device is called as Gannang's respirometer. So respiration coefficient measured by Gannang's respirometer, right? See here RQ means respiration coefficient. See, volume of carbon dioxide evolved, and during that respiration process, how much of oxygen is utilized? That is nothing but volume of oxygen consumed. So respiration coefficient value measured by the volume of carbon dioxide evolved divided by volume of oxygen consumed. So the value is called as RQ value. That one is also called as respiration coefficient value, right? So here we know we are eating carbohydrates, we are eating fats, we are eating proteins. These are the main source. Then we have to discuss about respiration coefficient values for different substrates. So here RQ values for the substrates. Here the first one is the carbohydrates. The very first one is the carbohydrates. We know our diet source is the carbohydrates, that is nothing but glucose. So here glucose C6H12O6, that is the structure of the glucose, right? And 6 molecules of oxygen is consumed. 6 molecules of oxygen is consumed, that is nothing but 6O2. And see, 6 molecules after the respiration process, 6 molecules of carbon dioxide is liberated and 6 molecules of water is liberated. Finally, ATPs, 38 ATPs production takes place. In that case, net ATP is the 36, right? So if we calculate the respiration coefficient for the carbohydrates, 6 molecules of carbon dioxide divided by 6 molecules of oxygen. So respiration coefficient value for the carbohydrates is 1. Respiration coefficient value for the carbohydrates is 1. So carbohydrates, respiration coefficient value is the 1. Next one is the fats. So here the example tripalmitin, that is one of the fat. Okay, we are taken here example. Here we are taken glucose example. Here we are taken here tripalmitin. So here totally 2C51H98O6. That is one of the tripalmitin structure. Within that structure, totally 102 carbon molecules are present, right? 98 hydrogen molecules are there and 6 molecules of oxygen atoms are there. Totally, for the oxidation purpose, 145 oxygen molecules are consumed. 145 oxygen molecules are consumed. So after that complete oxidation process, 102 carbon dioxide molecules are liberated, 98 water molecules are liberated. So if we calculate the RQ value for the fats or the tripalmitin, here 102 carbon dioxide molecules, right? 102 carbon dioxide molecules. Here oxygen molecules are 145. Then if we calculate RQ value for the fats, the value is 0.7. So RQ value for the fats is 0.7. So based on the amount of carbon dioxide evolved, amount of oxygen consumed. Based on that, we are calculating that RQ values. And the last one is the proteins. Remember the proteins RQ value. The main standard value is the 0.9. But some of the books here given there is an average range 0.8 to 0.9. For suppose one of the bit RQ values for the proteins 0.7 is given, 0.8 is given, 0.9 is given, and 1 is given. In that case, we have to choose that 0.9 answer. 0.9 is the answer. So, proteins always less than 1. Proteins, the RQ value is always less than 1. So, that is about the RQ values for the substrates. And some of the substrates also there. And we have to remember that RQ values also. I will explain now, right? So, this is about that. Okay, carbohydrates RQ values, fats RQ values, and the proteins RQ values, right? Remember, carbohydrates 1, fats 0.7. Here proteins range 0.8 to 0.9, right? That is about that RQ values for some substrates, right? These are the some RQ values for the different substrates. See these examples. The values also very important. Fats, we already know 0.7 RQ value. Proteins 0.9, right? Glucose is the 1. We know the carbohydrates RQ value is the 1. See malic acid. This is the component we are seeing in the TC cycle. The RQ value is the 1.3, right? See based on that evolution means based on that the carbon dioxide evolved and oxygen consumed. Based on that we are calculating. So the malic acid 1.3, citric acid 1.33, oxalic acid, right? Here the 4. Oxalic acid, the RQ value is the 4. So what we are seeing these values in that one, the highest one is the oxalic acid. The least RQ value is the fats. 0.7, right? See, anaerobic respiration RQ value. This is the one of the important bit. Okay, one of the anaerobic respiration process is going on. Within that anaerobic respiration process, okay, the RQ value of anaerobic respiration process. See, different values are given. In one option, infinity is given. We have to choose that infinity because anaerobic respiration process never depends on oxygen. That's the reason why we have to choose here infinity value. So during anaerobic respiration process there is no requirement of oxygen. That's the reason why the RQ value of anaerobic respiration process is the infinity. Okay. So that is about that some of the substrates having respiration coefficient values. Right. Next one we'll discuss about the factors affecting the respiration. Right. Coming to the last topic in this chapter, factors affecting the respiration process. So we know in the last one, in the enzyme chapter, we discussed different factors affecting the enzyme activity. Here also, respiration is affected by the different types of factors. We have to discuss sometimes asking bits from this area also. See, the very first one is that temperature. See, low temperature, enzymes are inactive. Right? Low respiration process. Very high temperature, again enzymes are inactive. Low respiration process. There is an optimum temperature is there. The temperature is 20 to 35 degrees centigrade. Right? At 20 to 35 degrees centigrade, optimum activity performed by the enzymes. If the enzymes are more active, the respiration is more. Because glycolysis is enzyme dependent process. TCA cycle is enzyme dependent process. Electron transport chain is enzyme dependent process. So here the temperature always required optimum conditions. The range is here 20 to 35 degrees centigrade. So that is about the temperature. Coming to the oxygen. Remember, see, anaerobic conditions also respiration takes place, but the organisms are different. The bacteria, okay, one of the best examples is some bacteria are only grown in anaerobic condition. Because if the bacteria are exposed to the high oxygen levels, the bacteria will be dead. The case is different. For suppose one of the eukaryotic, one of the human being is there. We require oxygen continuously. Okay, see, inhibition of respiration process by anaerobic conditions. That is nothing but inhibition of respiration, lack of oxygen, right? That effect is called as Parcher's effect. That effect is called as Parcher's effect. So within human body, if oxygen is available, respiration will be takes place. If oxygen is not available, okay, that respiration means aerobic respiration will not be takes place. Aerobic respiration will not be takes place. Lack of oxygen, if the respiration is not takes place, right, that effect is called as Parcher's effect. That effect is called as Parcher's effect, right? That is about that role of oxygen on respiration process. Carbon dioxide. See, in a cell, more amount of carbon dioxide is present, low respiration will be takes place. Because more amount of carbon dioxide levels are there, automatically that carbon dioxide levels influences the oxygen distribution. So more carbon dioxide levels are there, definitely less oxygen levels are there. So carbon dioxide is more, oxygen is less, automatically affects the respiration process. Automatically affects the respiration process. So here the carbon dioxide, more concentration of carbon dioxide, less respiration will be takes place. Right? Salts concentration. See, sir, what is the link between the salts and that respiration process? We know that there is in cofactors are there. We know about the prosthetic groups and the coenzymes and the cofactors, right? See, different types of the salts are there, which are nothing but enzyme activating factors. Okay, some are metalloenzymes are there, or metal activated. Yeah, some of the metals activates the enzymes. Some of the metals activates the enzyme. So here what I am telling, a group of metal ions are there which are involved in the activation of metal ions. So here salts, which are nothing but some of the metal ions. Sodium activates the enzymes, chloride activates the enzymes, right? In the same manner, zinc activates the enzyme. So magnesium activates the enzyme, manganese activates the enzyme. So different types of enzymes are there. That enzymes, okay, activated by the minerals. So here more salts concentration is there, more respiration will be takes place. Means that is not too much of high level. There is an optimum level. More salts concentration is there, more respiration will be takes place. Too much of the salt concentration is there. In that case, okay, there is no chance for the optimum respiration. Again, the mineralotoxicosis means high amount of minerals are accumulated within the cells. It shows the toxic effect. So there is an optimum mineral concentration is there. That one enhances the rate of respiration. Enhances the rate of respiration. Coming to the next one is the hormones are there. Hormones. We know presence of hormones, rate of metabolism is more. Within the plants, indole acetic acid, right? See, this one is nothing but oxygen, gibberellic acids are there, cytokines are there. Presence of these growth hormones, okay, more respiration will be takes place because these are supporting for the cell division. Cell division requires more energy produced by the respiration process. Automatically, these are the phytohormones which influences the rate of respiration. So presence of phytohormones, more respiration will be takes place. And age, we know young age, more respiration. Old age, less respiration because of the enzymes. I already said so many times. People are very active at the young age because of the enzymes. Enzymes are very active. If enzymes are active, more respiration. So young age, more respiration will be takes place. Old age, enzymes are inactive, less respiration. So old age, less respiration will be takes place. So along with these factors, see, is there any wound? Is there any damage that takes place within the body? So in that case also more respiration will be takes place. Is there any tissue damage that takes place? Right? Wound healing process, also more respiration will be takes place. During wound healing process also more respiration will be takes place because wound healing is nothing but the cell division process. Cell division requires the high energy. Cell division requires the high energy. Energy is produced by respiration. So during the recovery or during the wound healing process, more amount of energy is produced. That is nothing but more amount of respiration will be takes place. So that is about this chapter. So these are the factors which affects the respiration. In the next class we will discuss about the new chapter, right? Mostly photosynthesis.